So we're talking today about aerobic respiration. What you have up here is a diagram that is also in your book. You don't have to draw this. But this is what we're going to be talking about today. There were 2 steps in photosynthesis. There are 3 steps in respiration. So just as an overview, the first step is called glycolysis. The second step is called the Krebs cycle. Some people get that confused with the Calvin cycle because they both start with a K. But the Krebs with a K is respiration, and the Calvin with a C is photosynthesis. And then the last stage is the electron transport chain. There's a lot of stuff there, and you will know this by the end of the week. But for right now, just take a deep breath and don't panic. All will be well. All will be well. We will get there. Here's your first slide to write down. Okay, so photosynthesis takes carbon dioxide and water, and by adding energy from sunlight, creates sugar. The sunlight energy has been transformed into chemical bonds that's in the sugar molecule. So the sugar molecule has high energy chemical bonds. And now respiration is the opposite of that. Respiration is taking apart a sugar molecule and letting the organism use the chemical energy stored in those high energy chemical bonds. Okay? So when you and I eat food, this is what our cells do with it. Okay? I had a pumpkin smoothie this morning. It was wonderful. It was wonderful. And there was sugar in the pumpkin, and there was sugar in the little bit of agave syrup that I put in there. And there was, there's other chemical bonds that have energy that my body can take apart from the proteins and things like that. But my body is currently doing this with yumminess from pumpkin smoothie. Sugar is broken down 1 carbon at a time. There's chemical energy between every carbon-carbon bond. And so if your body were to just destroy the sugar molecule and let it all fall apart at one time, tons of energy would be released, and it would probably not be able to capture it and make it into a usable form. So your body is very careful. Cells are very careful. God designed a process that is stepwise and very, very highly regulated. That takes the sugars off one at a time to capture the energy in the chemical bonds. Okay? 1 carbon at a time. And the waste products, when we're all done, are water and carbon dioxide. So it's the exact opposite of photosynthesis. Photosynthesis is water and carbon dioxide making sugar and oxygen, and here we have sugar and oxygen making water and carbon dioxide. So if you take the 2 equations and you just flip the arrow, that's all the differences between respiration and photosynthesis. Okay? Only, just like photosynthesis, there's a lot more to it than just these 4 things. Okay? Come on now. Oh, so frustrating. Advance the slide. Thank you. Okay, so here is a picture very much like the ones we started with. There's 3 stages to this process. Glycolysis takes glucose and breaks it in half. So we don't pull off a carbon off the end first. We actually take the 6 carbon molecule and turn it into 2 3-carbon molecules. Okay? So glycolysis, glyco, sugar, lysis, to break. This is sugar breaking. Okay? First step is to take a 6-carbon molecule, break it into 2 3-carbon molecules. And then this goes into the Krebs cycle. The Krebs cycle happens in the spaces between the membranes inside the mitochondrion. So the mitochondrion, that's an organelle that all cells have, and the mitochondrion is the machinery of the cell that makes ATP, makes adenosine triphosphate. Okay? The Krebs cycle happens in the spaces between the membranes in the mitochondria. And then it sends some of its chemical products over to the last step called the electron transport chain, which happens in the membranes of the mitochondria. So similar to photosynthesis, where there's membranes called grana or thylakoid membranes, and then there's spaces between the thylakoid membranes, all in a chloroplast. Same sort of idea. There's membranes here, and then there's a space between them, and it's all in the mitochondria. So there's a lot of crossover, a lot of similarity between these 2 that down. So the first step in the process is called glycolysis. And sugar, glucose, is a 6-carbon molecule. And your body takes the glucose, and initially it's a ring. Glucose is usually in a circle. And so your body splits the ring and makes a 6-carbon line, and then breaks that into 2 pieces. So we have 2 3-carbon molecules, okay? And the names of those 3-carbon molecules are pyruvic acid. So it turns glucose into pyruvic acid. It does make some ATP while it's doing that because it releases the energy of that one chemical bond that it broke. It had 3 on one side, 3 on the other. It takes the energy from that central high energy bond, and when it breaks it into 2 3-carbon molecules, it captures some of that energy and makes ATP. So you have just a small amount of ATP made during glycolysis. But what's happening is the Krebs cycle and the machinery that's going to go on to break these molecules apart further needs a smaller piece of sugar to work with, a smaller molecule to work with. So glycolysis just gets it ready to feed into the rest of the machinery. Okay. This happens in the cytoplasm. So the rest of the process happens in the mitochondria of the cell. This happens just out in the cytoplasm. This doesn't happen in any particular organelle. And all living things conduct glycolysis. There are some forms of life that don't go further. They don't do the Krebs cycle. They don't do the electron transport chain. They instead do glycolysis and then some kind of fermentation to try to get some more energy out of it. And we will look at those options tomorrow when we talk about the other kind of respiration with the yogurt and the bread. But for right now, we're looking at aerobic respiration, what happens to things that take in oxygen and have mitochondria, which is most forms of life. Okay, so this happens in the cytoplasm. It costs 2 ATP molecules to get started. So the making of pyruvic acid out of sugar takes a little bit of energy to get going. So the cell actually spends 2 ATP molecules to start the process, but at the end they have made 4 ATP molecules. So it's a net gain of 2, okay? Remember, photosynthesis also makes a little bit of ATP, but it spends it right away in the creation of the sugar. We had Crystal and Crystal was the P group. Who was ADP? Oh, that's right. Okay, so it was you guys that were joined by ATP synthase in photosynthesis. Same protein, ATP synthase, makes the 4 ATP here using the energy that we got by breaking glucose into pyruvic acid. Okay, and just like we had NADP as the hydrogen carrier in photosynthesis, there's a very, very similar molecule in glycolysis, but it's not NADP, it's just NAD. So the similarities between respiration and photosynthesis, there's tons of them. There's a hydrogen carrying molecule here, but it's NAD. And NAD plus brings hydrogen to the next step of the reaction. Okay? This does not require oxygen. So organisms that live away from the presence of oxygen can do glycolysis, which is why things like yeast and lots of bacteria that live inside stuff where there isn't free available oxygen, they can still do glycolysis, and then they go into fermentation as opposed to the rest of the process we're going to get to. Here's, you don't need to know this level of information, but just so you see pictures of what's happening. Okay, we have a 6-member carbon ring in glucose. It takes ATP for that to be reduced, and it changes shape a little bit. And now we have, instead of 1 carbon flying off and an oxygen in the ring, that oxygen is still in the ring, but now the carbon that was here is flying off as well. So we have carbon on one side, carbon on the other, and a 5-component ring. And then we're going to break this bond here, and we're going to wind up with carbon, 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 and carbon, 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 and we have 3 pyruvic acid molecules, okay? And it's during some of the things that they do with this that we get 4 ATP out of the process, and we load up our NAD with hydrogens, so we have NADH, okay? So there's some more, obviously more detail to glycolysis than what you need to have in high school, but when you take this college again, you'll get a little more detail there. Or if you go into any other advanced life sciences. So Krebs cycle is the next step. Okay, so we had a Calvin cycle in photosynthesis, and it was a complicated series of chemical reactions that one carbon at a time built sugar. And now we have the same sort of thing in reverse. The Calvin cycle is photosynthesis. The Krebs cycle is respiration. And this takes the pyruvic acid molecules and pulls the carbon off one carbon at a time. And every time it does, it captures some high energy electrons and it captures some hydrogen as well. And it uses those to make ATP, and the waste product, when it's all done, is carbon dioxide. So that's why that's what you exhale, because your cells are taking apart sugar. Thank you for demonstrating. Your cells are taking apart sugar and using it to make ATP, so you can go do the things that your life requires. And the waste product of the sugar being broke down as CO2, your blood takes that dissolved CO2 gas to your lungs and dumps it into the air spaces of your lungs, where then you go, and out it goes, right? So one carbon at a time it's taken apart. What it's really after are these high energy electrons and hydrogen atoms that sugar has been carrying along with itself. And it captures those and sends them to the next step, the electron transport chain. And those high energy electrons are going to be used to power ATP synthase and make a whole bunch of ATP. So there is some ATP made in this step as well, not a whole lot. Here oxygen is needed, which is why we call it aerobic respiration. Aerobic, aero, has to do with oxygen. So that's why exercise that makes you breathe hard is called aerobics, okay? And so aerobic respiration is respiration, the breaking down of sugar needing oxygen. That's why this is called aerobic respiration. The electrons that are freed up by the breakdown of pyruvic acid are captured by 2 carrying molecules. We had NADPH in photosynthesis. We had NADH in glycolysis. NADH is used as well in the Krebs cycle, but it has a friend, FADH2. FADH2 is cool because it gets to capture 2 hydrogens, not just 1. So this is a bigger taxi cab of hydrogen. It gets to have 2 passengers. Okay, so FADH2, NADH bring hydrogens to the next step, to the electron transport chain, and those hydrogens are carrying high energy electrons from sugar. Okay, we do make 2 more ATP molecules just in the process of the taking apart of the pyruvic acid, and so we've made 2 in glycolysis. We make 2 more in the Krebs cycle. This happens in the spaces between the membranes and the mitochondria, and that space is called the matrix. And no, Mr. Anderson is not there. This is the space between the membranes of the mitochondria. The space between the grana, or the space between the thylakoid membranes and chloroplast, is called the stroma. Here it's called the matrix, the space between the mitochondria membranes. Okay. So We've captured 2 hydrogens in glycolysis and their accompanying high energy electrons. We've captured a lot more hydrogen in the Calvin cycle with FADH2 and NADH and their high energy electrons. We've made 2 ATP molecules in glycolysis. We've made 2 ATP molecules in the Krebs cycle. And now we're ready for the next step, but I think I have a picture first. Yes, I do. So again, this is not the level of detail you need to know, but just so you've seen it. I have pyruvate, which is a 3-carbon molecule. 1 carbon gets pulled off. I capture a hydrogen and NADH, okay? It takes an enzyme here. Oh no, I'm just kidding. The 2-carbon molecule is called acetocoenzyme A. That's going to enter this cycle, acetocoenzyme A. Enters the cycle, and there's 2 of them going in, remember? So I'm going to pull 2 more carbons off, and I'm going to capture some NADH out of it, and I'm going to make an adenosine triphosphate, and then I'm going to capture some more hydrogens, FADH2, and this cycle goes around, grabbing more acetocoenzyme A, breaking it apart, and off we go. This spins round and round and round in these matrix in the spaces between the membranes of the mitochondria. Okay, and every time it turns, we make 1 ATP molecule. 2 have gone in, and so I make 2 ATP molecules every time the Krebs cycle goes around. And I'm making 3 NADHs here. I'm making 1 more up here, so I'm capturing 4 hydrogens with NADH. And then I make 1 molecule of FADH2, so I capture 2 more hydrogens there. Okay. And again, back to this picture. So we have now broken carbon and broken sugar into 2 3-carbon molecules. We've spun the Krebs cycle around and broken it apart more. And between this process and this process, we've captured high energy electrons and hydrogens, and we're sending them over to the electron transport chain now. There you go. So this is the big harvest. We only made 2 ATP molecules in glycolysis, and we only made 2 ATP molecules in the Krebs cycle. So if that's all that you could make per molecule of glucose, that'd be pretty sad, because your cell uses ATP a lot for lots and lots of things, and it needs lots and lots of ATP. And if it only made 4 ATP molecules per molecule of glucose, you'd have to eat a lot of food. But you don't have to eat quite as much food because the electron transport chain is this amazing thing that God designed to harvest every possible usable high-energy electron out of glucose and turn it into ATP, the energy for your cell. The first 2 stages made hydrogen carrier molecules. They bring those hydrogens to the membranes in the mitochondria. And the membranes there are called cristae. So that's not what people intend to name their daughters after, where they name them cristae. You're a little mitochondrial membrane. You look so cute. But that's what they're called. So cristae and the matrix sounds like a bad sequel. But the cristae are there, and they have these enzymes in them that take all the high energy electrons that have been captured along with the hydrogens and use those high energy electrons to make ATP. So again, tying it back to photosynthesis, remember last week that when the water molecule split apart, we sent the other hydrogen, which I think was being played by Andrea. Were you the other hydrogen that came and got Noah all excited? Yes. Okay. And Noah got all excited about the presence of a hydrogen molecule and made ATP. That's the same thing that's happening here, right? ATP synthase is in the membrane, and when all these high energy electrons are being brought to it by all these hydrogen carrying molecules, it uses that energy to make ATP. And it doesn't just make 2 or 4, it makes 32 ATP molecules for every 1 molecule of glucose that your body breaks down. So that is a huge yield, 1 molecule of sugar makes 2 ATP in glycolysis, makes 2 ATP in the Krebs cycle, makes 32 more ATP in the electron transport chain. So organisms that can use oxygen and can do this make a lot more ATP than organisms that cannot. So here's a picture of what we're talking about. These enzymes are all in the cristae, okay, in the membranes inside the mitochondria. And all of these carrier molecules, the NADH and the FADH2, they bring their hydrogen to the electron transport chain. And the hydrogens come zipping out. Actually, I'm looking at this backwards. The hydrogen comes zipping out of the place where they're all dumped. And as they come out, this spins this little machinery in here, and it makes tons and tons and tons of ATP. So for all the hydrogens that are brought, it makes a total of 32 ATP from 1 molecule of glucose. Okay, it's very, very cool. And so the design evidence in this is just amazing. You understand why people.
That thing and make it like steadily.
Well, so how can I help you think about this? There's a lot of hydrogen that's pumped into this little gap, and as the hydrogen wants to come out, it goes through this channel, and it's bringing electrons with it. It's bringing high energy electrons with it. So it's a flow of charged particles through this little gap, kind of like if you blow up a balloon and you just let it out a little small. There's a little stream of air coming out. Same idea, there's a whole bunch of hydrogen, and it's forced out through one little space. And as it's doing that, it's carrying charged particles, which is electricity. It's a flow of charged particles. And so that energy, that flow of electricity, essentially, powers the machinery of ATP synthase to make ATP. So it's kind of cool. It's kind of cool. That's the last slide. What questions do you guys have for me? Does this make sense to you? So going back to our big diagram, these arrows here, glucose comes in to the process. We make ATPs. We make 2 of them. And then this arrow represents NADH going to the electron transport chain. And then pyruvic acid comes into the Krebs cycle. We make 2 more ATP. And this arrow represents NADH and FADH2 going to the electron transport chain. And then the electron transport chain uses all of that hydrogen to make 32 ATP. Okay? You guys are good with this? Sweet. Tomorrow we will look at what organisms that don't breathe oxygen have to do. And we will get to eat them, which is fun. So that'll be fun. You get to eat things alive tomorrow. Yeah. Okay,
Wait, what?